Hello, everyone. Today, I would like to give you an introduction to the Stereo-seq transcriptomics data analysis. My presentation today includes 4 sections. First, an overview of Stereo-seq technology, then introducing the Stereo-seq transcriptomics solution v1.3 product. Next is the data analysis workflow and main outputs. And lastly, a hands-on software demonstration. I will spend about 20 minutes on the first 3 sections and another 15-20 minutes on the software demonstration. First, let's get to the Stereo-seq overview. Stereo-seq stands for the Spatial Enhanced Resolution Omics Sequencing. It used a patent chip embedded with DNA nanoballs, or DNBs, each only 220 nanometer in size with 500 nanometer in between. The DNB carries special barcode and the poly-T oligos to capture mRNA directly from tissue placed on the chip. With this design, Stereo-seq features a centimeter-level field of view and nanoscale resolution, enabling high-resolution spatial transcriptomics across large tissue sections. Stereo-seq supports a wide range of research areas. First, developmental biology. In our first Stereo-seq paper, published in 2022, we applied Stereo-seq to mouse embryos at different developmental stages to study the mouse organogenesis. Next, brain research. Stereo-seq has been used in several studies, including the macaque brain atlas, axolotl brain regeneration, mouse hippocampus projectomes, and next, cancer microenvironment, such as the liver cancer, cervical cancer, melanoma studies. And lastly, immunology and infectious diseases. For example, Stereo-seq has been used to study trophoblast invasion during placentations, as well as lung infections caused by SARS-CoV-2. The Stereo-seq is designed for ultra-high resolution, but how does it achieve single-cell resolution in data analysis? As I mentioned earlier, each DNB spot acts as a tiny capture unit, and here we take data from each spot as a single beam. We group these beams together to form larger beams. To give you an idea, beam 20 forms a square that's about 10 microns, which is roughly the size of an animal cell. Besides, we use self-development algorithm to perform image-based cell segmentation to get individual cells, which we call the cell beam. These are the several Stereo-seq products, including both spatial transcriptomics and multi-omics. Current transcriptomics are compatible with fresh frozen and FFPE samples, which are the Stereo-seq transcriptomics solution and Stereo-seq Omni. The data analysis for these 2 products are very similar. Today, I will focus on introducing the data analysis for the transcriptomics solution, but the Omni users can also learn the general analysis from it. Now, let me introduce the Stereo-seq transcriptomics solution v1.3, which is the newest version of this product. This is the experimental workflow. Briefly, it includes 6 major steps: sample preparation and imaging, permeabilization experiment and transcriptomics experiment, library preparation, sequencing, and data analysis and visualization. These are the related products for v1.3 solution. The permeabilization set is used to determine the best permeabilization time for your specific tissue type, and the transcriptomics set is for the real transcriptomic experiment. The chip captures mRNA using special barcoded oligos, and then cDNA is synthesized directly in situ. Next, we use the Stereo-seq 16 barcode library prep kit for library preparation. The library is sequenced on DNB sequencer platforms with the matching sequencing kits. Data are then analyzed with our software suite, including the SAW and Stereo-map. Here, I want to elaborate a bit more on the library and the sequencing reads. This diagram shows the library preparation process. The CDN is PCR amplified and purified for 2 rounds, and CDN inserts and the surrounding barcodes are then sequenced by the DNB sequencers. This is what the sequencing read look like. It starts with the sequencing adapter, followed by CID sequence, which is the special barcode, then a fixed region, followed by the MID, which is for gene count, and then the poly-T and the CDA inserts. On the other side, we have the sequencing adapter as another sequencing adapter. The read one sequence 50 BPs for the CID and MID, while the read 2 sequence the CDA insert for 100 BP. We have compatible sequencing kit for DNB sequencers. Those kits, along with the sequencing strategy, determine the sequencing type used in the data analysis. Okay, now let's get to the data analysis workflow and outputs. This is the general workflow of the Stereo-seq data analysis. It starts with the image quality control using Stereo-map, followed by reads analysis and image processing in SAW. After that, we use Stereo-map again for basic data visualization. If the image from the SAW needs further adjustments, we can manually process them in Stereo-map and then rerun the SAW with the corrected image. That covers the standard analysis workflow. For more advanced downstream analysis, we rely on community-developed tools. Now, let's look at the main tools used in analysis. First, we have SAW. This is the command line tool that handles the core processing steps. Next is the Stereo-map, a desktop software used for image quality control, manual image processing, data visualization. For downstream analysis, we choose the right tools for each analysis step. Some commonly used ones are StereoPy and ScanPy in Python, and Seurat and Spotlight in R. Today, I will mainly focus on the STAN analysis workflow, so I will walk you through how to use SAW and the Stereo-map. So what is SAW? SAW, which stands for Stereo-seq Analysis Workflow, actually includes several pipelines. The main one for standard analysis is called count, which handles the core processing to generate expression data. It also has several supporting pipelines, including macref, checkGTF, realign, reanalysis, and convert. Each is serving a specific purpose to help with the data preparation or reanalyze. These are the key steps in the standard analysis by SAW. We start with the input data, which goes through reads pre-processing, reads mapping, annotation, and counting to obtain the spatial gene expression matrix. Next, the image is lined with the gene expression data to obtain the registered image. Then we perform tissue and cell segmentation and extract gene expression data based on those segmented regions. Finally, SAW performs clustering and differential gene expression data analysis to reveal the spatial patterns in the data. In this scheme, we can see the key inputs needed for the pipeline. First, we need a chip mask file, which comes from the Stereo-seq chip itself. Next, we have the sequencing reads generated from the library. We also need the image data, which come from staining and imaging the sample. On top of that, we need a reference genome chosen based on the sample species. Finally, we have some experimental parameters that also needed for the SAW run. Once all the input files are ready, the good news is the entire pipeline runs only with just one command. Then we can see the software system requirements for running SAW. It runs on Linux environment with at least 8 cores, though 24 cores and more are recommended. You'll also need at least 128 GB of memory and 1 TB of free disk space. You may be wondering how long to run SAW. As a reference, here are examples from two mouse brain samples, one stained with ssDNA and the other with HE. For those runs, we process 1.8 billion reads and 1.6 billion reads using 24 cores. Each run took about 8 to 9 hours. Now, what is Stereo-map? Stereo-map is a desktop software for image processing and visualization. It has three main modules: Visual Explorer for data visualization of spatial gene expression data, and region selection for further analysis. Image processing for manual image adjustment, and tools which include the image QC utility. Stereo-map is compatible with both Windows and Mac system. For both, it requires at least 16 GB memory, and we highly recommend using SSD storage for better performance. Now, let's see what the output files from a standard analysis look like. The key result files are stored in the outs folder. Inside the key, you can see that the feature expression matrix are stored in the feature expression folder, and with the main format to be GEF or GEF. For secondary analysis, like clustering and differential expression analysis, the results are saved in the analysis folder, and the files are in H5AD format. Also, we have HTML, report, and visualization files for quick review and data visualization. Here, I'm going to introduce them a little bit more. This is the report HTML file. On the summary page, we can view the gene expression heat map and the registered image to quickly check if they are aligned properly. Besides, there are key statistics like total reads, MID count, gene numbers, and the sequencing saturation curve for you to quickly access your data. The report also has clustering analysis result along with the marker gene analysis. They are very useful information for your downstream analysis. Next is the visualization file. They are used for data exploration in Stereo-map. You can quickly visualize the expression of target genes, check clustering analysis, and select regions of interest for further analysis. Now we reach the last part, the software demonstration. I will show you a step-by-step tutorial how to use Stereo-map to export data analysis result. So let me exit here and enter my local Stereo-map software.
Okay, this is the interface of the Stereo-map, and on the left you can see the three modules here. To explore or visualize the Stereo-seq data results, let's choose the first one, Visual Explorer. And I already have the output file on my local computer. And this is the visualization folder, decompressed from the visualization tar.gz. And let's go inside. These are all the files required for visualization. And to open them, let's select the file with the stereo suffix and then confirm. And this is our data. It's a mouse brain sample that is fresh frozen and stained with ssDNA. And first of all, we can scroll up and scroll down to zoom in and zoom out. And on the top, we can see the 3 drop-down boxes. They are for the gene selection, the image layer, and analysis layer, and also the bin size selection box. And for the current image, it's the bin 50 at the bin 50 bin size. And we can see that this is a gene expression heat map. And let's scroll up to see what the spot bin 50 look like. Now we can switch to bigger bins. This is the largest bin 200. And now we can switch to the smaller bins. Overall, we can see that bigger bins can give you better contrast, give you a stronger pattern than a smaller one. As the smaller bin or smaller spot, they only have like limited genes or gene expression in it. Okay, so that's the interface. And next, let's check the image registration. And to do that, let's go to the layer. And we can see there are 2 layers selected. One is under image layer, which is ssDNA, and the other is under the main analyze layer, which is transcriptomics gene heat map. And to check the image registration, we want to see if the 2 images align well. And currently, seems that the gene heat map is on top of the ssDNA, and we cannot see the others very well. And we can adjust it by go to the gene heat map and change the transparency of the heat map so that we can see the other image clear now. So by doing that, we can do a rough check and we can see the edges of these 2 images. They overall align pretty well. But you may wonder how to do it in a precise way. And to do that, let's use the track line tool on the top. This shows the Stereo-seq track line template. And now we want to do is to see the ssDNA, the track line from the ssDNA image, if they matches with this track line template. And now let's go to the layer again, and let's dim the heat map. So only the ssDNA is showing up, and to let the track line from this image show, we have to adjust the normalization, as the track line from this ssDNA image is very light. And now let's do the normalization adjustment. So we can see the thin track lines here, and they matches very well with the template. So that's the precise way to check image registration. Now let's go back. And let's do a quick check on the data. I'll look at a few brain marker genes here. By the way, as a reference, I use the Allen Brain Atlas as a reference. So by clicking on the patient, the name you can find where those regions, this like a structure located in the structure. For example, this is outside the peripheral part is the isocortex, and this part is the hippocampus, and another one, yeah, that I will use later is the fiber tract, which also has a name called white matters. Now let's start by checking the first marker, which is a common neuronal marker called neuron or RBFOX3. And let's search for it in the gene selection box. Now we can see the distribution of the RBFOX3 seems pretty widely expressed in the whole brain. But we can play with the beams to see a stronger pattern. Let's switch to beam 100. Now we can see a bigger contrast. It's more highly expressed in the isocortex region and also the hippocampus region, which makes sense as those regions have the highest neuronal densities in the brain. And next, I would like to check another neuronal marker, which is the dentate granule cell marker. Should be located here. It's called PROX1. And let's click on it. Remember to deselect the previous one as we want to check the single gene now. And what you see is they express in this dentate gyrus region, which is the expected result. And next, I want to check an oligodendrocyte marker, which should locate mostly in the white matter region. It's called the PLP1. And it aligns very well with the white matter regions. So all these marker genes shows the results match our expectations that give us confidence in the data. Next, you may wonder how to check multiple genes together, as we only focus on single gene at one time, right? So to do that, I'm gonna use another marker for the white matter. It's called MVP. Now these two genes are both selected, and these are the genes. This is the gene expression heat map of these two genes, right? And to see them separately, let's go to layer under the gene heat map. We can see on the bottom there's a display schemes. Let's click on the multicolored instead of heat map. Now we can see these genes are assigned with different colors. We can click eye icon to see one or the other. And they seem have a strong co-expression pattern. And we can also change the color if we click the color icon here. So overall, they align very well. So this is how we check multiple genes. Beyond the gene expression, you may want to know what the clustering results look like. And to do that, let's first go back. To find the clustering, let's go to the, let's use these two drop downs. For the Stereo-seq V1.3 analysis, we have clustering result for bin 20, bin 50, and cell bin. So now let's click on the bin 50, and you can see the cluster and UMAP show up right here. And now let's click on the cluster. You can see that these are the clustering results mapped into the brain context. Overall, on the right side, we can see there's 27 clusters, and there are a few layers in the cortex region, which align well with the brain structure quite well. And now let's try the cell bin. And this is the result for the cell bin clustering. And on the right side, we can see there are 28 of them. And let's zoom in. So this cell bin or the single cell shape, like they use the single cell shape here. And how to get it? How to obtain the single cell or segmented individual cell? As I mentioned earlier, it's done by the image-based cell segmentation. And now let's see that layer from the image layer. And that's the cell mask and cell mask adjusted. This stands for the nucleus border identified by the ssDNA image, which labels the nuclei. And then this cell mask adjusted is the real cell border that is based on this nuclei. And let's take a look at them. And we can dim the cluster layer so we can see the original ssDNA. And let's click on the cell mask. So you can see that this is the border of this fluorescent image, which is labels the nuclei. And let's check the cell mask adjusted. So they are the border expanded from the nuclei. And that is the single cell shape we see in the cell bin. The cell bin is very useful if we want to perform single cell level spatial analysis. And let's continue. And then lastly, if you want for spatial analysis, you have your regions of interest already, and how to perform the region selection in the stereo map. Now I would like to show you the lasso function here. And let me use an example. Say we are interested in the hippocampus region, and this is the dentate gyrus, or DG, and this is CA. And I'm interested in comparing these two groups and to see what are the differentially expressed genes are. And to do that, let's first select the regions. Click on the lasso. Let's do the selection for the DG now. Let's follow the instruction, which press the control, and the other hand use the mouse to circle the area. And if you don't do it well, you can remove those regions. Okay. And now let's save it. You can see the save icon right here. And let's give it a name. It's called target group and a label as the DG. And let's do the same thing for the CA region. Okay, now this is the CA region, and let's save it, put on the same group and give a different label. The next thing here is to save these regions. Let's go to the groups. You can see that the select regions show up here. It's under the target, and there's two groups, and there's all. And let's click the dot here. You can see the GeoJSON for differential expression. Click on that. It asks me to select the method. I want to do a label versus label. And the others, I just leave like that. And let me save on my local computer, say in the lasso folder. And now it's successfully saved. The next step is to integrate these regions to the SAW secondary analysis. I already completed analysis, and now let me show you the result. It's a gene list for the differential expression analysis. It's in CSV, so let's use the open icon on the left, load a CSV file. Under my secondary differential expression, and you can see it's right here. And you can see that the first column is gene name, then the log4 change p-value for one group, and log4 change and p-value for the other group. And they sorted by the p-value. And from the top, they are the most significant differentially expressed genes for the first group, and on the bottom, they are the most significant genes for the other group. So let's check them. And let me click on this gene name. Let me exit lasso first. Let me click on the gene name. And now you can see that these are the gene expression of this gene, of this LRRTM4. And let's try bigger beans to see the patterns. And overall, it looks pretty strong. That is significantly expressed in the DG region compared to CA. And let's check the second one. Remember to deselect the previous one. And we see the same pattern. It's highly expressed in the DG. Now let's try the other way around. So click on the bottom one. And this is the gene expression for the lingo one, and you can see that it's highly expressed in the CA region. Okay, so that's how we explore the spatial differential expressed genes. That's also the end of the demonstration. Hope it gave you some idea how to explore and analyze the Stereo-seq data.